Oh, okay. We are now recording. Everybody, so why this is such an important day is this is the day we talk about gram positive and gram negative. You might remember this is, I'm using two PowerPoints. I'm using this one, and because depending on where you're looking, if you haven't loaded it or at least take a look at it, I'm going to use the other one here from that Anderson, which I call Nestor, chapter 3. It's about 60 slides down. They have a lot of lab stuff early in that particular sequence. And that's one probably I'll be using the most because I don't love the illustrations. I don't mind talking about it, but I don't love the illustrations. A quick catcher up that I probably forgot to tell you about because I don't recall it when I was talking to the 11 o'clock group. I all of a sudden like, oh, I should have told these guys that. Uh, we talked about positive and negative movement or taxis. And so, in the context they gave you was chemo, which is by far and away the most common. They move either toward certain chemicals because chemicals get in the environment. They diffuse and they touch receptors and spins the flagella one way or the other way, either attracting or repelling. It's not magic. It's there. But there's other kinds, and we don't see it quite as much as chemical movement. Photo for light. There's photosynthetic bacteria we really don't deal with so much in this course. So they call that phototaxis. They're either attracted, if they're photosynthetic, they want to go to light. If they're not photosynthetic, they might move away from light. Magnetotaxis, some really odd bacteria that we really don't study, that don't really interact with us, have tiny little iron-like, I don't know if exactly iron or what the metal component is, but basically magnetic fields move them. So they're odd. Air is pretty easy to understand. We're going to get to that when we start talking a little bit about where they grow and how they use air in the different categories. Some move toward the air, some go away from it. You'll see that in the lab. When you start to grow stuff in the lab, particularly in tubes, when you inoculate tubes. Did you inoculate tubes to see if they grew any stuff in them yet? Okay. You're going to see, depending on what it is, some species will grow, like the Bacillus subtilis will grow on the top because it's aerobic. And some of them will grow all throughout the tube, and there's different ways. And we'll go over kind of and dovetail that into lab. But that has a lot to do with either they want air or they want to get the heck away from air as far as possible. And then heat, the same thing. We're going to see bacteria, like most of the bacteria affect us, enjoy the temperatures that we basically around our body temperature. But there are ones that do better in cold and one do better with a little warmer. And I'll have a whole little presentation about that. Got through that pretty quickly, which leads us to this. And it's just, I never liked the diagram. The only thing I really liked about this was the thickness of the peptidoglycan compared to how to seeing the complexity and the limited amount of peptidoglycan here. That's the way I looked at it. So I'm going to go back to this one and show you the Nestor and work with that because I like the way they present it. Really, the idea of attacking what makes the cell wall is what holds it in shape. If you break down the cell wall, that whole envelope doesn't have security, and changes in the amount of fluid surrounding it will either cause it to shrink or burst and effectively destroy the bacteria. Most often it bursts because it goes into the area where there's effectively too much water by way of concentration. And there are two types. And this will be, did you talk about this in lab? The folks about it yesterday? Okay. Maybe that's next week. Gram positive and gram negative. You'll learn the stain about that. Did you do some staining in lab? Yeah. So you did what's called basic staining. Basic, it has to do with the chemistry, not so much about being a basic skill, although both terms might apply. So gram positive and gram negative. The way that we have to distinguish that right away, what kind of infection is. So they're giving you some video, some little things. This is what a gram stain looks like. You'll see this in lab, and effectively you'll see like blue, purple versus kind of red, pink, depending on the quality of the stain. And basically it allows you to distinguish different kinds of cells. And as you can see, when it's in that purple area, the blue-purple area, I was just talking about Bacillus with the big B, Staphylococcus, Streptococcus, thick peptidoglycan. These teichoic acids, as you will see, kind of anchor it to the membrane. I mean, something has to secure to the membrane. It's porous, so stuff can get through it. There's no outer, so you can refer to this, there's no outer membrane, and we're going to get today at length what they call the LPS, or lipopolysaccharide layer. That's an oddball. We only see it in gram negatives. That means it's unique, big word, to gram negative, and it's always a toxin to us if it gets where it shouldn't be, even though our gut is filled with gram negative bacteria. But understand that in the colon, it's really not inside of us. It's just, they're just on top of the lining, actually helping us. That's when you have like a burst appendix or something like that. Oh boy. And all of that gets in the abdominal pelvic cavity and big trouble, called peritonitis. And yeah, this is all about penicillin. We'll look at that. I'm not too worried about the lysozyme, the stuff we use in the lab. And then E. coli, Neisseria, which is gonorrhea, Pseudomonas, which you're going to get sick of if you're working in a clinical environment. Ugh. And it don't smell good either. Okay, all of those things are part of it. Okay, so we have, relatively speaking, the thin layer of peptidoglycan, and you're going to see it's sandwiched between a couple of membranes. They don't have the teichoic acids. Okay, and we'll get to it. The whole difference, okay, when they talk about porin proteins, think pores are where moisture gets in and out. Again, that big thing in peptidoglycan is porous. So you don't need to make channels. It's already got the channels inherently based on the structure. Where because you're going to see membrane on either side, we have to put pores in. If you want to think of a living thing, not we, but a living thing has to have pores in there to allow water-soluble things to move effectively in and out. And I mean, and that's a really good overview. And this is peptidoglycan and why I use it. So I'm sure this again is the learned LTF or learn to forget. It's a structure, and I know it's complicated looking at that. I tried to draw it schematically over there on the board basically, but maybe the room is a good example. The bricks here, these sections here, they are the glycan. Glycan is a polymer. The little N thing at the end means it's basically a sugar with nitrogen. Nitrogenated sugars are what living things use for structural material. You studied connective tissue, all those wonderful, there's collagen, which is a protein. You looked at the ground substance and all that stuff, right? Most of you remember you had things called glycosaminoglycans that were in there. Those are basically, glycans are biological structural material. Depending on the species, so they give it strength. So they're like these bricks. Now, if you want to make a brick wall, you put the brick here, put a layer here, put a layer, put a layer. These are nicely fitted. But if you pushed hard enough on a brick wall like that, it would fall over. Because you didn't have anything holding it together. That's the peptides here and here. Peptides, these tiny amino acid sequences, basically are the mortar, for lack of a better term, that holds the bricks, and that's what gives it its strength. And indeed, again, it's got porosities to allow water-soluble things to get through. What is it constructed of, this polymer? Polymers don't always have to be one thing. Like we learned in A&P, we talked about glycogen, we talked about starch. They were all made out of glucose, one repeat unit. This one's got two. We just call them NAMs and NAGs because I would not ask anybody to remember N-acetyl uramic acid or N-acetyl glucosamine. See, I knew it. That's fine, I get to show off. NAMs and NAGs. They've alternated. So polymers have repeat units. This has a 2 molecule or 2 unit repeat unit. So just say alternating NAMs and NAGs, okay? That's the backbone, so to speak. And it's held together by these peptide groups. Particularly, they call these tetrapeptide chains, interbridges, peptide chains, and multiple layers. And gram-positive, 30 to 40. Folks vary between 20 to 40. 30 to 40 for me is the most common area. Again, you can see they look like a carbohydrate if you remember your chemical structure, but they've got nitrogen. Glucosamine is important. A lot of people take, as they get to my age or younger, they'll take glucosamine with MSM. You'll see that in supplements all over the place. Okay, you may know folks in your family who take that for arthritis. It's sort of it functions as a building block, they think, for cartilage. People will say, oh, it doesn't really work. I think it works. Just telling you. It doesn't work for everybody. That's why it's not universal. But this is one of the supplements that really does. If it doesn't help you, stop taking it. It helps you. So, alternating series of NAMs and NADs, glycan chains, tetrapeptide chain links them together. Peptide interbridges, particularly in gram-positive cells, because they're able to sort of keep those bridges aligned and these large stacking of them. So that's the first thing that you have to understand. It's not the same. Gram positives, super thick. Gram negatives, 1 or 2 layers of peptidoglycan. So already you see, if you look at it closely, something that attacks the peptidoglycan is going to already have a potentially limited effect on gram negatives, the much more complicated cell wall. So here it is, relatively thick, depending on who you read. It's got what are called teichoic acids. They really hold things on that are their basic stains, okay? Notice what protrudes from bacterial cells, acids. And the stain, if we're using methylene blue in lab, basic stain. It's basic because the part that stains, the colored part, we call the chromophore, is basic in chemistry. Basic stains are attracted to acids, you know? Voila, that's why it latches onto it. See, you learned something today. Okay, you didn't learn. So it has, basically, there's some gel-like material, don't worry about it. Basic cytoplasmic membrane, phospholipid bilayer. Yes, you have pores in that because, again, water-soluble things have to get in and out. You don't need it so much here. Okay, I can handle this one. Okay, maybe not. You would not like to meet that in a dark alley. It's not. That's a bad-looking cell wall. That's entirely too complicated for me. This is a thin layer, 1 or 2. You can see here a peptidoglycan. They don't have the interbridges, but we're not asking you about that. And the thing that makes it special, unique, is the outer layer of the outer membrane. The peripheral, remember, central and peripheral. The outermost layer of the outer membrane, it's this guy. So you have a phospholipid, it's like a giant sandwich. Membrane inside, membrane outside. It's interesting, if you can break down the membrane, you've got a win. And you can expose the peptidoglycan. If you can somehow get through the membrane, it's a win. If you go over to SCAFE today, for the class that just preceded you, they're going slightly crazy. They have a giant farm test. They're going absolutely nuts. Okay, this is part of this part when they're learning about antibiotics. That's why this stuff is important for you to remember. This is going to come back. Normal membrane, phospholipid bilayer. Then you have an outer phospholipid layer here on the inner leaflet of that outer membrane. But you got this guy who's totally different. The so-called lipopolysaccharide layer. It's not a phospholipid. It's a lipid with sort of a uniquely arranged sugar. Okay, that's there. And in the middle, you've got 1 or 2 layers of peptidoglycan and an interesting fluid. Another layer of fluid is the wrong term. Fluid/gel called periplasm. A lot happens in the periplasm. A lot of activity goes in there. Bacteria, magically. This is a single-celled organism, but it's developed ways where antibiotics come in and has little what we call efflux pumps to kick them out. That's where some of the resistance comes from. Pretty clever for a single cell. I wouldn't have thought of that. You're not that smart anyway. So voila, what makes that outer so-called lipopolysaccharide layer? You have lipid A. That is the toxin. It's something totally foreign to us if it gets in what we call sterile tissue. And so other than gut linings are on the surface, everything else inside is sterile tissue. So it's a problem. It has a polysaccharide, they call the core polysaccharide, and they have something called the O antigen. Some people call it the O polysaccharide. It's variable and it's used for identification. Down the road, you're going to hear about this. There's about half dozen really bad strains of E. coli. It will say E. coli O157H7. So, frankly, when you look at E. coli under the microscope, they all look alike. They're little red classic looking rods under the microscope. Relatively easy to stain. Really comparatively easy to identify. But how do we know which is which? These markers are able to be traced chemically. So we're able to test this E. coli in the laboratory. The O157 means we can identify that particular marker. It doesn't necessarily imply that it's more dangerous. It just so happens this one is. The H marker, the marker that comes from flagella, is on the flagella, so they have variations in that. And the O is on that O, for lack of a better term, the O. So when you see that, don't be confused. We do that all the time, and you're going to see a lot of it in micro. We're going to start talking about flu. You might hear H1N1, the one that's kind of going around now, H3N2. H and N have to do with specific proteins that play a pivotal role in either how it gets into you or it gets out of you and infects other cells. So a lot of this has a great deal of significance. So the so-called LPS, or lipopolysaccharide layer, or outer layer of the outer leaf, is basically so foreign that it basically triggers your immune system. It doesn't need a lot. This is the most confusing thing. When I put this on the test, students just, whatever, they can't grasp it. I tried to identify it a little earlier. Bacteria generally hurt you, not all. But if it's a broad brush, and I don't like to do that, by secreting or giving off toxins. And all bacteria do it. For long-term, called exotoxins. And they happen in both gram-negative and gram-positives. The so-called endotoxin means it's not a secreted particle. It means it's inherently part of the bacteria. Which means it's only, big word, we don't use a lot of onlys in science and medicine, because they're the only ones with that lipopolysaccharide layer, and therefore they're the only ones that have that lipid A component. So in general, we refer to the lipid A component specifically, or the LPS layer generally, and yeah, it can be deadly. Again, ruptured appendix, tumor in the genitourinary or digestive tract causes a breakdown in one of those hollow tubes, and all of a sudden, that's all the stuff that's in there spills out into the abdominal pelvic cavity. That's peritonitis. Gunshot wounds, stab wounds. Those are the kinds of things you see those, and it's like, oh, we got trouble. And depending on where you're going to, where many of you, and I know I'm speaking to people who are going to do clinically. Unfortunately, you see a lot of this stuff, depending on where you are. So large amounts of this in the bloodstream, and immediately your immune system reacts to it. That's the lipid A, that's the immunological component, and this is the identification, the O antigen. Most of the texts kind of forget about that H antigen, but it is related to flagella. And I mean, just keep that for general knowledge. I'll probably just only ask you about the O antigen. I had a student contact me more than once so far about, I am going to do the review recordings. The other, it's okay. You have to pay for them this time. I'm joking, but it would be nice. Okay, 75th birthday, May 10th, coming up. Could you start thinking about it? I'm terrible. Begging for gifts. What? So you gotta write that down. Nope, I know. And inevitably it's Mother's Day. See, I get short shift. Oh, they're celebrating and my birthday. Oh, it's Mother's Day. My birthday. But we'll be, and they were supposed to have a big deal. My son, I think, did I tell you him being promoted to 06? We're going to do that here in Pittsburgh. That's a big deal. So we're going to do it here in Pittsburgh. We're going to do it then, but for whatever reason, it's a week later, right around graduation. I'm trying to get him to do it up in the chapel. We'll see if I can get lucky. Moving on, and then I can invite you guys. Yeah, see, and for bonus points, you would all come. Go team! Don't mess this up, etc., etc. Okay, moving on. So all of that, I mean, I can't emphasize. There's going to be a half dozen questions easy on a test about this stuff. If nothing else, that should embed it in your mind. The outer membrane, including particularly antimicrobial medications, small molecules and ions, water, things dissolved in water, can cross because of they have porins. Okay, and effectively that's the best name. They're transport proteins or porins. There's just so many different names that we use. And I'll review on the way I reworded on the test to make sure you're aware of it. And secretion systems important in pathogenesis. So not only is it stuff in, it's stuff out. So not those exotoxins that make them dangerous. What makes that O157H7, okay, is it secretes a nasty. It's not because of the lipid A. It's because of a nasty toxin that is extraordinarily damaging, and other bacteria have that toxin that are there, and it's potentially deadly. When you hear about deadly E. coli outbreaks and people dying, this is by far and away the most common one. I think we first identified it at I don't know if it still exists, there was a fast food chain called Jack in the Box, and this one was up in the state of Washington, and what happens is, like, if you don't, the meat they get, if it has fecal contamination from, it's not butchered and ground properly, you get like intestine stuff in there. And if you don't cook it sufficiently to kill it, then you can spread that. Just saying. So, and we'll go into a lot of that stuff. Between cytoplasmic membrane and outer membrane is the periplasm. Okay, exports proteins and accumulate unless specifically moved across the outer membrane. So proteins get in there that are made inside the cell, and then it has ways of pushing them out. There's different kinds of transport systems I don't get into. I mean, if you're going into microbiology as a field, you learn about these very sophisticated mechanisms. That's what I was saying. They have a lot of mechanisms, and we look at antibiotic resistance, we'll revisit these that are there. And the whole idea, why peptidoglycan, it's only in bacteria. So if we can interfere with peptidoglycan in any way, shape, or form, most likely it will burst. And that's what penicillin does. Penicillin interferes with the assembly that I drew up there, and you saw before with the PowerPoints. It doesn't allow the glycans to chain together. More effective in gram-positives, easier access. There's more of it. The outer membrane, the gram-negative, blocks it. So when we studied, just to give you a hint, we began to know about this when they started using, they only had penicillin, they used it for everything. And immediately they saw it wasn't so effective against gram-negative, so they added an amino group. And that changed it chemically so it could get through the membrane a little bit easier. We have a name for it, aminopenicillin. You might have heard of it called ampicillin, or the form that works orally, amoxicillin. So we'll get into like the family tree of penicillin. Okay. But that's in our third unit. I'm just trying to give you some context for this. So lysozyme, something we have in our saliva, and something that's found inside a lot of cells. Okay, tears, saliva, other body fluids, inherently they're protective. They destroy that peptidoglycan, and again, more effective against gram-positive. So it may make you want, we have mechanisms that protect us against bacteria that are there. And this is the gram standard. I'm not going to go over it. That's lab. Not every bacteria, they all have peptidoglycan. It's not always organized completely. And this is when you heard the term pleiform that has funny shapes. These are funny shapes. This is called mycoplasma. Okay. Penicillins don't work. lysozyme doesn't work because the peptidoglycan doesn't really play a role in stability. What plays a role in the stability, and you can see it here, and it's an important term going forward, is sterols. You all had AP1. What stabilizes our cell membranes is a sterol. All eukaryotic cells have it. Cholesterol. Not a lot of bacteria. These are an oddball bacteria. This is called, and really the best species to understand this is this mycoplasma. The names can be confusing. Okay? Mycoplasma, the most famous one of these is called Mycoplasma pneumonia. Respiratory disease, also known as atypical, because it ain't like regular pneumonia. And way you've heard about it, called walking pneumonia, and it's very common in college students. It's pretty easily spread. It's like you'll go to the health, whoever does the health care or urgent care or emergency room, say, "Oh, you have walking pneumonia." And I mean, you're not sick like a pneumonia, like you can't get out of bed for a week. It's like we're talking low-grade fever, cough. It persists for a long period of time, but you can still function more or less. So in the military, recruits who lived in barracks easily spread, a la dorm rooms. Okay, that's how it spreads. So mycoplasma pneumonia, also known as walking pneumonia, and you can't use penicillin for it. So thankfully, things like the Z-Pak and tetracyclines work very, very effectively against it. We're not doing the archaea, less well studied, similar but not the same. The S layers, we don't have to worry about any of that stuff. Okay. So that's this side. I'll go over here now because I want to stay with the one we've been using because I think that you follow it a little better and it gives you the ability to kind of follow it in a textbook type of thing. So here it is, and you can see, and they've done a pretty good job. I mean, they have the distinctive color. You can see there's no teichoic acids here to anchor it. It doesn't have to be anchored. It's basically a membrane sandwich, a peptidoglycan sandwich on membrane. Pull the mayo. Bad joke. I thought it was didn't work, did it? Yeah, mission. There's a wonderful. I just finished watching it. 2-part series. It takes about 4 hours. Mel Brooks, the 99-year-old man, just came out on HBO. It's hysterical. God, what a wonderful individual. Lord willing, he will be 100 in June. So peptidoglycan here, and you can see the pores that they have, and they'll talk about porin proteins, and that'll be the best term to use. And some of these proteins are channels and things like that. So you can see it fairly clearly. So it's not a terrible diagram. It just doesn't help me. So yes, it's about the shape of the bacteria. Okay, strong structural support to keep the bacteria from bursting or collapsing, and most often it bursts. And this has to do with fluid movement or osmotic pressure. One of the things when you have an infection, your body sends white blood cells, and it also sends a lot of fluid. It dilates blood vessels. You'll learn about that a little bit in immunology, and we'll talk about it a little bit here in response to disease. And again, that's in subsequent chapters. And so it leads to lysis or disintegration or rupture of the cell. And again, they're showing, and again, I didn't like this as clearly as the other ones. That's just me. So I don't want to use that particular slide. It's thick in nanometer terms. Different kinds of, I just use the term teichoic acid. Maintenance and enlargements contribute to the acidic charge on the cell surface, part of what makes staining possible, and why gram-positive, you will learn in the lab, are easier to stain than gram-negatives. Gram-negatives will pick up some of the stain, but not as effectively. You have to do something different to that. I don't want to get into the science behind it. The gram-negative, relatively thin, more susceptibility to lysis. Okay. They're going over the techniques. You can look at these as well. And so I thought the other presentation was more thorough, at least in my opinion. Now, notice the difference. Mycoplasma, this is called mycobacteria. And nocardia is something we don't deal a lot with. So it contains peptidoglycan. These are gram-positive bacteria, but it's mostly lipids. It has a capsule. It's made of mycolic acid. It's a unique capsule. We have a couple of really famous ones. You'll even use one in the lab. Got it. And again, mycoplasma, these are called, genus is Mycobacterium. Famous members of that, closest. Another one you don't see very much. I've seen one case of it began leprosy. When you use the lab, just write the emphasis gets higher. Well, and SMEG, that's what I'm saying. Another one we're going to come up down the road and start talking about tuberculosis, big M bolus, because it comes from cows. It has to do with immunization that's used in certain countries. Or why is it such a pain to deal with? Number one on the hit parade is the mycolic acid capsule. It's enormously thick. It's very hard to stain. The stain we use is called the acid fast. Again, it ain't fast, but we fasten it to the acid capsule. So a lot of times it's like fixing carbon and fixing nitrogen. We're affixing. In this case, fast doesn't deal with speed, it deals with attachment. So this is a stain you'll do in lab, along with an endospore stain and things like that, most likely. And it isn't contributed, it's really a signature piece of why it's bad. Very resistant to chemicals, very resistant to antibiotics, very. Multiple antibiotics are needed, tuberculosis, all the time. And so archaea, atypical, we're going to skip it. Mycoplasma and other cell wall deficient bacteria. Here's the distinction. So read questions carefully and read the answers. Is it mycoplasma? Somebody who's trying to trip you up like me might have mycoplasma versus mycobacterium. See? Don't look at me like that. You're going into nursing? They eat their young. So all of those things, walking pneumonia, L forms. We'll clinically, if we get to them, we'll take a look at it. They're probably, we're talking mostly about chlamydia is pretty interesting. When we get to genitourinary disease at the very end, I think it's kind of like this. So gram-negative, the lipopolysaccharide layer, a signaling molecules and receptors, and it's an endotoxin. Okay, special membrane channels in the outer membrane. Within that, and of course, the regular cytoplasmic, all the typical stuff that we use. Energy reactions, nutrient synthesis, gatekeeper, stuff going in and out, selectively permeable, all of that stuff. Standard phospholipid bilayer biological membrane. So the outer membrane, the gram-negative, contributes an extra barrier, resistance to a certain and quite a few antimicrobial chemicals, more difficult to inhibit or kill. Don't mistake, oh my God, there's a gram-negative. You're going to see a ton of gram-negative infections. I can't even tell you how many bacterial cystitis, okay? Basically, in the female, you see a ton of bacterial bladder infections. They are relatively easy to treat with antibiotics. Okay? So don't broad brush and say, oh my God, it's gram-negative. Gram-positive, the infections you're going to see the most are gram-positive, and ones like MRSA or MRC, which we'll get to, methicillin-resistant Staphylococcus epidermidis, they're bears. So you can't just say, oh, this is gram-negative or this is gram-positive. If you're going to tell me in a lab it's harder to stain a gram-negative, I'll buy that. But I don't equate those things with pathogenicity. Okay, so for instance, alcohol dissolves the outer membrane. That's why I don't think the alcohol swabs are terribly effective, but they do get rid of, mechanically, they get rid of a lot of surface or microbes. Yes, if they have an outer membrane, that will affect them. Yes, with something we'll learn about called an enveloped virus, they work on. You'll be just as far ahead using soap and water. I got news for you. But we all get in the habit of doing it. Trust me. So you always follow these things. When you get into aseptic technique, you continue to do it. Okay? You must have to be very watchful. Treatment of infections caused by gram-negative bacteria require drugs that can cross the outer membrane, of course. Yeah. Who cares? Yeah. Cytoplasm. See, now we finished the cell wall and it only took us 35 minutes. Aren't you glad you came today? Not seeing a lot. See, thank you. Some people. Water, sugars, amino acids, salts, a bunch of stuff. Okay. Now, so there's not a lot to say about cytoplasm. This is interesting. The bacterial chromosome. We use the term nucleoid because it's an oid. It's not a nucleus. It's like a nucleus. It looks like a nucleus. Erase this. Okay, I'll erase this. If I was only a little taller, this would be easier. So if you look at our nucleus, the bacteria are arranged in pairs, linear pairs. The DNA is regular, double-stranded. Down the road, when we get to viruses, you'll hear something about single strands and stuff like that. But linear and in pairs. Another story, one from mom, one from dad. Now, in a bacteria, just use a genetic rod, you have effectively a giant circle of DNA same DNA, double-stranded, and we call this supercoiled. Supercoiled is like if you took a rubber band and you twisted it and you twisted it and you twisted it, you know how it kinks up and all that stuff? You compact it. That's what supercoiling does. I mean, how big are we? Our DNA has somewhere between 35 to 50,000 genes. E. coli, how tiny is an E. coli? That you can barely see with 1,000 times magnification, has about 10,000. There's a lot of DNA. When it explodes out, you'll see this. It can wrap around an E. coli about 1,000 times. That's supercoiling. Very thin strands, like that. So that is called the nucleoid, the region where we see this. It's like a nucleus because it's sort of in one place, but it doesn't have a membrane. And a lot of these have, hopefully the color works. Oh, look at that. A small circle called a plasmid. When I say small, maybe you're looking at 20 to 50 gene markers. And plasmids, by definition, non-essential pieces of DNA, but useful, typically conveying good things like resistance to antibiotics. And we'll get into how they're passed genetically. But for the time being, plasmids, drug resistance, toxins, enzyme productions, would be good or bad, honestly. And plasmids have, as you will see, the ability to move. And they can form and they can disappear. So non-essential, and it's interesting. You'll see that when conditions are adverse for bacteria vis-a-vis the surrounding environmental conditions, sometimes plasmids will form. When the conditions improve, indeed they improve magically in that population, they'll start to disappear. So they're very, but most bacteria have at least one and typically more than one. So they're really important parts of this discussion. And because of the distinction, again, it's a circle of DNA. And that, I can't emphasize enough how important that is, because when we start looking at genetics, the beauty of genetics is we have pairs of chromosomes. One's dominant and one's recessive. Both dominant or both recessive. So you always have, and we only express one side. We have a lot of inherent genetic mechanisms to prevent mutations. Bacteria mutate a lot. hence MRSA. They mutate all the time. They're inherently genetically unstable. And anytime you put something adverse in their environment, like an antibiotic, they tend to mutate 1,000 times more frequently. How's that? To the point where they debate in certain environments, 1 out of 10 bacteria will mutate. We make billions of cells every day. They rarely mutate. You know, a handful will mutate out of that billion, and we have an assortment of mechanisms as well as a great surveillance system in our immune system to gobble them up and get rid of them. Very interesting. Love that. So much fun doing this. So we'll get more into the mechanisms. We'll explore that in a little bit more detail. I'll tell you about when the time comes. I think I have to add a tiny little component or an extra lecture about something. So ribosomes, just like us, protein synthesis, except they're just free. They're in the cytoplasm. They have 2 subunits. Okay, they're mostly ribosomal RNA and protein. All ribosomes are like that. The difference is because we have a small and large subunit just like they do, except when they come together, it's not like you're adding them, it's a density thing. When they combine, they have a different density. So it isn't just a sum of those S units. We just use S. So they combine that way. In bacteria and archaea, there's 70S. In ours, they're 80S. We will get to, when we look at eukaryotic cells, the big difference. The fact that we have 70S in our mitochondria. Our mitochondria are interesting because we've got little circles of DNA and 70S ribosomes. What's that all about? So here you can go 50 and a 30, happen to make a 70. Oh, who cares? I'm not going to go into chapter and verse of all, we're not going to do protein synthesis and different parts of it. And then just to complete the picture, as I do have enough time to get through this, food storage. So they don't have the capacity other than in what we call inclusion bodies, and we use a lot of them, okay, for identification. So that, again, bacteria that need air float to the surface. They have vesicles that might have gas, for instance. So we can identify they are. The crystals or the iron oxide, the magnetic ones, some of them are fairly exotic, okay? They have tiny micro apartments, okay, for certain but it's not a big deal for us. But I'm saying we use most of these things more for identification in the laboratory, and really, if we're really doing advanced looks at it, you look more at the details of physiology, so to speak. And again, we used to think that they didn't have a cytoskeleton. It's like you went to the grocery store, you went to Giant Eagle, and you had about a half dozen things, you threw them in one of those plastic bags, and that was a bacteria. Stuff just kind of, you could shake it up and they'd still all be in there. Not so. We know there is some kind of cytoskeleton. It contributes to the shape. It's generally under the membrane. Okay. Down the road, that's something that they might address, because you're always looking for differences or things that are interesting to attack vis-a-vis trying to control disease. Like I said, we're running out of antibiotics. We're always looking for new ones, and we're looking for other methods to deal with them. First time, not enough time. I'll introduce it. This is the next big chapter. So, gram positives, gram negatives, peptidoglycan, the distinction between the nucleoid, 70S versus 80S ribosomes, all of the above, really important factors test-wise. The other big thing are endospores. I'll show you an animation when we meet next time. I just want to introduce it here. For our purposes, we're only going to see them in bacillus, the one you're going to do in the lab because it's aerobic, and Clostridium, all that, the tetani, the botulism, blah, 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 blah. C. diff, that you're going to see. This is the single most confusing thing. We call it a vegetative cell. If I tell you someone is in a vegetative state, what does that say to you? We have a permanent vegetative state in this patient. They're laying there, kind of like you do in class. Not a lot. See, I got a laugh out of that one. For those of you that were awake to appreciate it. That if you and I are vegetating, we're in big trouble. On the other hand, sorry, how to do that. That was from an old time guy named Soupy Sales. He said, "You can count on me. 1, 2, 3." Guy's name, he was from Tennessee, and his name was Milton Hines. So they nicknamed him Soupy because Hines made a lot of soups back in those days. Nevertheless, bacteria are like plants. That's how we thought they were microscopic plants. So when they're vegetating, they're replicating. They're happy. I vegetated all day. Good for you. I had a great day. So when you hear the term vegetative, they're active, they're multiplying. Some bacteria, principally Bacillus or Clostridium, can form effectively a dormant form, almost a state of suspended animation that it forms when conditions are adverse, and they effectively live forever. Okay, they're called sporulation. It's all about environmental conditions. It's all about available nutrient. They are endospores. They resist extremes of heat, drying, freezing, radiation, and chemicals that would kill vegetative cells. They are really a pain. And like I said, they're everywhere. They're in the carpet. They're on your shoes. They're in your house. They're in your dorm room. They're everywhere. Can't avoid them. That's our next topic of study. And there's really good animation, and I'll point it out and how the spores form and what's unique about them. Thank you very much.